Let's start the module 2, which the first part will be you will see that there is a little bit different names where in your syllabus it says endomembrane, and endomembrane is nothing where these proteins are going to be synthesized, and then these are going to get moved into specific membrane organelles, and it's the same thing. So this is basically protein trafficking. So the first part, we are going to just do the 13.1 and 13.2, and 13.3 just by itself, we will have that on Tuesday, and probably we're going to spend some time over the exam. That's the reason I just kept the 13.3 separate. We know that we do not have that much of time, but I'll try to manage. So 13.1 is the targeting protein to and across the ER membrane, where you are making some soluble proteins, which will be in the lumen of the ER. We're going to talk about that, and insertion of the proteins in the membrane, which are the integral proteins. We'll talk about those in the section 13.2. So the first thing that what we are looking at, the overview of the protein trafficking, that you already know from your basic idea that you have these polyribosomes. They are making the mRNAs, or reading the mRNAs, making the polypeptide. As soon as the polypeptide is done, it will extrude or go out of the ribosome, and if it is a cytosolic protein, it will fold itself in the aqueous environment, and that's it. It will remain in the cytosol. Say, for example, glycolysis happens in cytosol, so all these proteins of glycolysis are present in the cytosol. On the other hand, you have all the different organelles. You have nucleus, chloroplast, mitochondria, endoplasmic reticulum. If the protein needs to go there, they have to have an address which is embedded in them. And if you look at this, like here, there is the small pinkish sequence right here, and depending upon that sequence, the protein will be targeted either to the nucleus or the chloroplast, mitochondria, or it may go into the endoplasmic reticulum. So that's the overall idea about this. So we are not going to talk about in this class or in the course the targeting to the nucleus, intermembrane space, to the mitochondria or the thylakoids, whereas we are going to look at the bigger picture where they will be moved to the endoplasmic reticulum, rough endoplasmic reticulum, because we are going to see that the ribosomes are going to be moved on top of the endoplasmic reticulum. From there, these proteins are going to transfer into the Golgi complex, and from the Golgi, either they will be going out of the cell or they can be directed to the internal part of the cell, like a lysosome or peroxisome. Now, this process by which the ribosome synthesize the proteins and the ribosome get attached to the endoplasmic reticulum, where the protein is either made in, or the mature protein is either extruded inside the lumen, or it goes to the membrane, no matter what, they will then be transferred to the Golgi to be transported in a proper way. So think about that. The endoplasmic reticulum is your assembly line or the factory, and the Golgi complex is your distribution center. From there, it will be distributed. And these pathway, not the nucleus, mitochondria, or the chloroplast, the pathway through the endoplasmic reticulum and the Golgi and the distribution is known as a secretory pathway. And the problem is that the name is a little bit of a misnomer. You think that these proteins are going to get secreted out of the cell, which is not. So many of these proteins can be secretory proteins, which will be secretory out of the cell, but many of them will be directed like a lysosome, peroxisomes, many other organelles or structures inside the cell. But the idea is that secretory pathway involved the endoplasmic reticulum and the Golgi. If they involve the Golgi and the endoplasmic reticulum, that's the secretory pathway, and that's what we are only going to talk about that part. We are not going to talk about the targeting to the specific organelles, because this is the bigger picture. So just a little bit idea about the largest organ, endoplasmic reticulum. These are, I'm not going into the detail. These are very self-explanatory, just how they look like. Smooth endoplasmic reticulum is much less. They make mainly lipids like hormones, testosterone, estrogen, all the steroid hormones. And just have an idea that in a three-dimensional structure, this is the rough endoplasmic reticulum, and in the middle part is a smooth endoplasmic reticulum. So all these proteins are going to be made into this rough endoplasmic reticulum. They will be moved into the smooth part, and from the smooth part, a vesicle is going to bud off, and this will travel to the Golgi, and then they will coalesce with the Golgi, and this is what you call as a cis face of the Golgi. If it's like this, I mean, like, if you just remember from your basic idea that you have structures like this, so this is called the cis face of the Golgi, and this is called the trans face of the Golgi. So the cis face is where these budded off structures from the endoplasmic reticulum is going to go fuse. Then they're going to get sorted, and then they are going to go to the trans face. They're going to cross, and they're going to go into the trans face of the Golgi. From there, they are going to get distributed. So that's the overall idea. So now the problem is that how you study these proteins that are being synthesized and manufactured in the endoplasmic reticulum. So it is almost impossible to get an endoplasmic reticulum because it's so large and it's intricate network, but you have a huge advantage to the cell biologist. What you do that you take the cell, no matter what, from the tissues or the cells in culture, you homogenize them. All these endoplasmic reticulum transiently is going to break apart, but immediately they will go to seal and produce something what you call as a microsomes. So this, you can see that from this mixture of smooth and rough endoplasmic reticulum, you can get the smooth and the rough microsome about 100 nanometer in diameter. If it is rough, you will have the ribosomes on top of them. The interior is biochemically exactly to the endoplasmic reticulum. So this is what you isolate, and you study those. And there is absolutely no problem of isolating these. It's a very standard procedure to do this. So just some pictures of the rough and smooth endoplasmic reticulum. Smooth doesn't have any ribosomes. Rough does have. So how you do this? You do the density gradient centrifugation. We have already talked about that. When you layer them on a gradient of sucrose, the smooth endoplasmic reticulum doesn't have the ribosomes. Much lighter, will float towards the top, whereas the rough will be much towards the bottom, and you can just isolate the rough and the smooth endoplasmic reticulum. So for all our purposes, we are interested only to the rough endoplasmic reticulum, which actually are responsible for making the proteins. We are not interested in the smooth, so the whole of the topic will only talk about the rough endoplasmic reticulum. So here is one idea. The signal hypothesis. This was proposed a long time back in the 70s, 80s by Günter Blobel, that how the proteins find, or the ribosomes find, the endoplasmic reticulum to make the protein go inside the endoplasmic reticulum. There must be something. And this was the idea that there must be a signal in the protein or the polypeptide that is being made by the ribosome that helps the protein to go to the ribosome. Not to the ribosome, that makes it go to the endoplasmic reticulum. So the idea is that the polyribosome is making from the mRNA, making the protein is in green, and it does have a red portion at the beginning. This is called a signal sequence. It's a specific set of amino acids, which is just like a zip code, and depending upon that, the proteins use that zip code to destine it to the endoplasmic reticulum. And it has already been shown then that after it goes to the endoplasmic reticulum, this signal sequence is cleaved off by an enzyme called signal peptidase. So it is cleaved off, and the mature protein goes into the endoplasmic reticulum's lumen right here. And then the structures which are responsible for this dissociate ribosome get separated. We're going to talk about detail about that, get separated. So here the idea is that there should be a signal. The hypothesis said that there should be a signal in the protein which allowed to do this. And this idea was proposed 70s, 80s, and then finally this was the experiment which was done by a German-American scientist, Günter Blobel. So what he did, that he made an in vitro translational experiment where you have the ribosomes, you have the mRNA, and you just add a specific labeled amino acids, which is labeled with a radioactivity. You will see that in your book, it says pulse chase experiment, where you can do it with the whole cell as well. So read that. I'm just not explicitly saying that, but when the protein is being synthesized, it will be labeled by that. So you can you can identify that. So it will be radioactive, just the protein. So when this is being synthesized by the ribosome in a test tube, he added microsome in one sample and he did not add microsome to another sample. What he found that when he ran these proteins in a gel, now remember that when he's taking these samples, the ribosome and the microsomes, he's just mashing up all of these protein and denaturing them and then loading onto a polyacrylamide gel electrophoresis SDS page. Remember that if you are not familiar with that, this is your prereq material. So this is your protein gel electrophoresis, and according to the molecular weight, towards the plus end, the lower the molecular weight, it will be towards the bottom of the gel. Higher the molecular weight, it will be up there. So when you add these samples, there will be hundreds of proteins, but remember that as you have added during the translation, the labeled radioactivity, only the protein that is being made by that mRNA by the ribosome, that is the only one is going to get labeled, none other. So you can easily identify that by you take the gel, you put an autoradiographic film in that, and you will have those black bands on the autoradiograph. So what has been found, that if you have the microsomes, the protein size is larger. If you do not have the microsomes along with your translational mixture, you have a smaller size protein. So the idea is that there must be a signal peptide that has been cleaved from this, and this must be in the endoplasmic reticulum. So this is today's standard. This is a very basic experiment that was done. But again, nowadays with all the sophisticated techniques, the signal hypothesis is known, proven. Unfortunately, it's still called signal hypothesis. It's a fact. It's not a hypothesis anymore. He received Nobel Prize in 1999 for the work of this. So it is known that they have the signal which makes the protein go into the endoplasmic reticulum. So how do we know that where these proteins are destined to? See, here is another experiment where you take the polyribosomes in vitro translation, and after you do translate the proteins, you add microsomes to them. And it was found that at that condition, all these proteins are outside the microsome, because here, if you just purify the microsomes and look for the proteins, there is none. On the other hand, if you make an in vitro translation where during the translation you add the microsomes in there, the microsomes were found inside the microsome. So this gives you another idea that a signal is involved which lets the protein go inside the microsome. Another example of this, so this is the idea that the protein that is being made on the ribosome, so the blue thing is the mRNA and this pink thing is the protein coming out. When microsome is present, this protein goes into the microsome. This cannot happen without the microsome during the translation. If you translate the protein beforehand, after that you add the microsome, this does not happen. So what the idea here, what I'm trying to say, that the movement of this protein into the endoplasmic reticulum need to happen simultaneously as the protein is being translated. So the movement of the protein inside the microsome is known as a translocation of a protein. So this is another important term. We are going to constantly use that. So translation from mRNA to the protein, translocation, movement of the protein from the ribosome or polyribosome inside the endoplasmic reticulum. That's your translocation. You're moving the protein. So how we prove this? That the N-terminal signal peptide is cleaved off by the signal peptidase within the microsome. We have seen that. But let's take a look at how we can prove that this is actually the case and the protein is inside the microsome. So why we are doing this experiment? Once again, see that this experiment tells you that it is associated with the microsome. We really do not know whether it's on the membrane, it is inside, or it is on the surface. So here, whether they are sticking to the surface or not, we cannot tell that from this experiment. But to have the idea that they are really inside, we need to do another experiment, or the scientists did another experiment. Same thing that you can allow in a cell, the protein synthesis to happen with the polyribosomes. all these proteins are in the lumen, you break up the cell, homogenize, and immediately you will form these microsomes. And these microsomes should have proteins inside them. So you now add in that mixture of microsomes protease. And after the treatment, you isolate the microsomes once again, and when you look at the protein, they are intact. They are not digested because they are being protected by the microsomal membrane, which is the endoplasmic reticular membrane essentially, and the protease cannot penetrate that membrane. So they are definitely inside, okay? Now, how you prove? You have the second layer of proof that if you just add a detergent to that microsomal mixture and add the protease, these proteins are going to get degraded. That means the detergents are going to make holes into the membrane. We already know from your previous chapter. So they are going to make holes into the membrane with the detergent, and your protease is going to digest the protein. So from this experiment, it is now clear that all these proteins are inside the microsome after being synthesized. So that is clear. And then again, another proof of that, not going into the detail, that the proteins also become glycosylated by enzymes within the microsome. This you can also look at different molecular weights. The molecular weight due to the glycosylation changes. So if you take the protein and you add an enzyme which will digest the carbohydrate, you get a lower molecular weight compared to that. So all these different types of experiment proved that is these proteins with the signal peptide in there are destined to the endoplasmic reticulum, and it goes into the lumen of the endoplasmic reticulum. That's what we're going to talk about in the first part. Remember that we are still talking about the lumen of the endoplasmic reticulum. In the second part, we'll see that they are also directed to the membrane of that. But as we are only talking about the proteins which are inside in the lumen, we are emphasizing that. So here is how a signal sequence really look like. This is being proven over and over, because if you just cleave that, the protein becomes a cytosolic protein. It is not going anywhere. So you can easily mutate a protein to understand that the importance of the signal sequence. It is also known as a leader sequence, start transfer sequence. These are some of the idea. The sequences are not conserved. There are many different types, but there are some features which are conserved. Most of these signal sequences will have an hydrophobic residue, so which is green, and they may follow by a positively charged or negatively charged residues. And it's not always the case. There could be, but that 5 hydrophobic residue should always be there. That's the signature part. And then it could be only plus sequence, minus or plus sequence, plus or minus sequence followed by that. That's your signal sequence. Here, just an idea of typical sequence for importance to the ER right here, for the import to the ER. This is the sequence that we already saw. For other, there are some other sequences as well, and we are not going to go into detail, just to have an overview of that, how this signal look like. So let's do the first clicker question. Which of the following proteins will not have a signal sequence? Proteins targeted to cytosol, rough ER, nucleus, mitochondria, chloroplast. Stopping it now. Good, everyone? Stopping it now. Good. That's the answer. The proteins targeted to the cytosol. Just a few of you chose rough ER or the nucleus. Remember that the signal sequence is an address. If it doesn't have the address where it is synthesized, which is a cytosol, is going to stay there. So that's the idea of that. This is the very first picture that you saw. If you remember that, that's what it is. Okay. All right, so let's move on. So the A, that is the correct answer. So the next part, what we are looking at is how these, now we have an idea that you have a signal sequence and these proteins are going to get transferred into the endoplasmic reticulum, and how this actually happens. So what happens that once this secretory sequence, hold on one second. Yeah, okay. So once the protein is synthesized, it will have an N-terminal signal sequence. And this is actually hydrophobic. You have seen from the sequence, mostly hydrophobic in nature. So as soon as it comes out of the ribosome, it does have an unfavorable environment because it's cytosol. So immediately what happens, there is a class of protein which are known as an SRP, or signal recognition particle. It's a huge, large protein complex. It contains 5-6 different proteins and the RNA closely associated with the ribosomal RNA and the ribosome. And immediately what it will do, that it will capture and sequester the signal sequence. That means that it will not allow that signal sequence to be exposed to the watery surface because it's just going to quickly cover that. And now that SRP, or the signal recognition particle, is going to pull down all these polyribosomes towards the SRP receptor, which is on the ER membrane. So as soon as the signal sequence is bound to the SRP, here you can see that the green SRP is bound to that purple sequence, and the SRP will bring it down to the SRP receptor. That receptor is on the ER membrane. And now you see that the polyribosome, as it is synthesizing, the polyribosomes are moved on the surface of the endoplasmic reticulum. This is the overall process. So how the structure of SRP, very complicated. You do not have to remember any name of these proteins or the RNAs. A large structure. So the P54 sequence, that's the only thing that we are looking at, that this is one of the proteins. And there are lots of these methionine residues that binds to many of these signal sequences. And these are mainly hydrophobic in nature. And just to give you an idea that what happens. And again, do not have to go into any detail. This is the P54 homolog from FFH. That's archaeal, bacterial archaeal. These are all bacterial archaeal homologs of the P54. And this is the hydrophobic binding groove, where the hydrophobic amino acids that are in the signal sequence bind, and this whole particle brings it down to the receptor. And one of the important thing, that this is the most important part, the SRP needs the hydrolysis of GTP, or the power of GTP, to do this movement. It is not ATP, it is GTP only. So SRP is always GTP, and here what you are seeing, that there are two monomers and the GTP is bound. This is an active form of this is an alpha subunit. It is an active form. In this process, it can bring the polyribosomes along with the signal peptide to the endoplasmic reticulum. If they hydrolyze the GTP, that means they are released from when the GTP hydrolyzes, this SRP is going to released from this alpha beta receptor, and they're going to recycle, charge with the GTP, and again bring the next one. So this is the cycle, and the GTP hydrolysis is involved in this process. So here we are looking at the next process, that, okay, all right, SRP brought that material, now what? So now what happens, that you have the SRP receptor, once the SRP binds to the SRP receptor, this protein, which is just bound to the SRP, is now moved onto another protein which is known as a translocon, and it is gated. So upon the signal sequence binding to the translocon, it's going to move the signal sequence inside the translocon lumen, or the channel, and the gate is open. So now the protein is moving towards this channel, and as it is moved, the signal peptidase, which is present in the groove as well, or very close, is going to cleave the signal sequence, and the secretory protein is going to deliver inside the lumen. So that's the process of this transfer. So again, the two important factors, you have an SRP, binds to the signal sequence, brings it to a SRP alpha beta receptor, transfers the protein to the translocon, and translocon is the channel, is a very large multi-channel protein, multi-pass protein, which transfers the protein into the lumen of the endoplasmic reticulum. So now you have the folded, mature, soluble secretory protein. So all the secretory protein which are soluble and that are not membrane bound, they are free in the soluble condition, say lysosomal hydrolysis that needed to go into the lysosome, or a protein that's needed to be secreted out of the cell, not membrane bound. All of these protein are going to get into the lumen and get transferred to the Golgi for the direction. So this is the first part of this idea. So how you understand, and again, I think that your book elaborates this, but I will just stop that how you know that this translocon is transferring this protein. So we'll just go quickly because this is very detailed in your textbook. So there is no reason for going into too much detail. So you synthesize mRNA, which is about 70 base pair, how the experiment was done. 70 base pair long mRNA, which is just the N-terminal part of the prolactin. And then this is a Sec61 alpha, and this is isolated from the yeast. And you are doing an in vitro translation. And when you are doing an in vitro translation, you add a specific type of lysyl tRNA. And this lysyl tRNA have a modified lysine residue in there, which with the presence of a cross-linker can do some cross-linking reaction. So what happens that in the middle portion you have a lysine residue, and you allow the translation to happen, and as translation is being happening, you have the Sec61 alpha in the in vitro mixture as well, and the protein is getting passed into this this structure. Now the important part is that what the scientists did, and this is the most clever part, that when to stop this process, because if you cannot stop the protein, you really do not know where is the protein and how fast it is passing and going into the lumen of the endoplasmic reticulum. So this mRNA did not have a stop codon. So as it did not have a stop codon, the protein will get stuck with the ribosome and it's not going to get released. So that was the clever experimental part that they did. So this protein is stuck with the ribosome. Now what you do after you allow it to translate, you shine a bright light which will cross-link the lysine to the translocon. And now you purify the translocon, and along with that, the peptide was also found with the translocon, which proves that that Sec61 alpha is a translocon component in the yeast. Just a little bit of structure of archaeal Sec61. This is another one, and it is gated, double gated. You can allow only the peptide to move in. When the peptide is moving in, or the polypeptide or the protein, this particular structure moves out. Also, there are some isoleucine hydrophobic residues around this area, which is like a gasket and doesn't allow the small molecules to pass through when the protein is translocating. Otherwise, there will be other problem. It only allows the polypeptide or the protein that is being synthesized to move into the ER lumen. So here is another just rendition of the whole thing, just summarizing that whole idea that your ribosomes is synthesizing this signal peptide first. As soon as the signal peptide emerges, it's going to bind to the SRP complex, and this SRP complex is going to bound to the SRP receptor and transfer that structure to the translocon component. As the translocon component is moving the protein into the lumen of the endoplasmic reticulum, the signal peptidase is going to cleave the signal peptide, and the whole mature protein in the folded form is delivered. That's your secretory protein. So good, everyone, with this part? So this is the first part of that, but there is an exception. And again, remember this for your quiz question. So the idea is that so far what we have seen, that here, as the protein is moving to the translocon, the ribosome is sitting exactly on top of the translocon. So basically, there is only one way for the protein to go. It can only go in one direction. And where is the power of the protein to go through? The translocon don't use any kind of energy. It is just the push from the translation, which is of course energy dependent. Just the push of the translation pushes the protein into the lumen. But the protein cannot go in the opposite direction. The protein only can go in one direction because it is plugged on one side with the ribosome. But there are some exceptions. There are some proteins which are synthesized in the cytosol. This is in yeast. There are some example in higher eukaryotes as well. And these proteins are synthesized inside the cytoplasm. So if we now go back, that which protein will not have a signal peptide, I have to add a line that there are some exceptions. I have to add what is the general thing. Do you understand that what we are saying? Let's go back to that one. So the concept is maturing, and that's why purposefully I did that. Up to this point, which of the following proteins will not have a signal sequence? Proteins targeted in the cytosol. Now we are telling that there are some targeting proteins in the cytosol will also have signal sequences. These are exceptions. So if I ask this question in your exam, I have to give you a much more specific type of question that there are except some of the exceptions, like in yeast. Otherwise, this question will be very wrong. So here, what we are seeing that these proteins are synthesized in the cytoplasm, which will have a signal peptide, and it can move in through this. But there is a problem for this process. It can find the translocon to move this, but as there is no plug of the ribosome sitting at the top, there can be a back and forth movement. The protein is trying to move in, it can slide out readily. So how you can manage this process that the protein, as it is finding the translocon to move into the endoplasmic reticulum, it is not backsliding out into the cytoplasm. So to do that, there is a particular protein which is known as this binding protein, or BIP. This binding protein is a chaperone. And again, a chaperone protein is a helper protein. It helps the protein to properly fold, properly do their function, and to mature. These are the general rule of the chaperone. And it is a family of HSP70. You do not need that, but if you read your textbook, you will see that. It is a heat shock protein, 70 kilodalton, and this is known as a binding protein. As long as you know it's a binding protein, that's all you need. That extra information is not important. The concept is more important. So this binding protein will hydrolyze with the help of this Sex63 complex. It will use the ATP hydrolysis, and it will transfer an ADP onto the oncoming protein. So now you can see that the oncoming protein chain is like a bead of ADP on top of that, and that will not allow the protein to slide back. So this is the process by which the addition of these ADPs are going to make the protein go in only one direction. And this BIP protein is also a unique protein that after the protein is completely inside, and it just takes a few seconds for the protein to slide into the endoplasmic reticulum. Once the whole protein is inside the lumen of the endoplasmic reticulum, BIP automatically hydrolyzes and takes up new ATP, and the whole cycle goes on. And with the Sex63 complex, it transfers the ATP to the ADP. It transfers the ADP to the incoming polypeptide. So this is the process. So overall, BIP is involved. ADP beads, that doesn't allow the protein to slide back in. That's the process. So you have some post-translational translocation. These are some of the exceptions in yeast, and there are few examples, not many, in higher eukaryotes. So take-home message, most of the proteins which are secretory soluble proteins, which are in the lumen of the endoplasmic reticulum, most of them are transferred by the SRP on the membrane of the endoplasmic reticulum. So translocation happens along with the translation. So translation and translocation is similar. Translocation is co-translational as well. So I'm just making this word once again, but again, remember that most of the time, translation of the protein and movement or the translocation of the protein happening at the same time. This is normal, and just few exceptions are there where the translation happens in the cytoplasm, then the protein is moved. So that's it. I repeated quite a bit of times because once you get this concept, it will be easier for the next part. So the next clicker question will be, which of the following protein complexes can bring the translating polyribosomes to the ER in absence of GTP? SRP, SRP receptor, translocon protein complex, none. So D is the correct answer, which is none. But 30% of you translocon protein complex does not bring that. It is already in the ER membrane. It doesn't go anywhere. So just remember that. Let me explain that. Well, not the. Yeah. So let me close this one. So let's go back. So here you can see easily right here, it is clearly written in this one that SRP needs that GTP hydrolysis. If you go back right here, we talked about that GTP is involved in this process. This is the only particle that will bring the polyribosome in this process, as we said, that to the endoplasmic reticulum, which is the co-translational translocation. That is the only thing that is involved. On the other hand, if you can see that in the yeast, there is no specific protein which is bringing that protein. It is the signal sequence is finding that translocon by itself, and that helps the movement of the protein. So GTP is not involved, and none is the correct answer. All right. So next question. Is it possible to synthesize a secretory protein and bring it to the ER without bringing the ribosomes and involvement of SRP? Some people pressed C. Really? Somebody woke up. Yeah, so yes is, of course, the correct answer. And that was just the last slide right here. So this is the correct one where there's no involvement of SRP, and this is the correct answer. All right, so all good. Ribosome and involvement of SRP. So the next portion is that moving proteins onto, yeah. That's a good question. Yeah. So let me just go into, not that way. So basically you have the signal sequence right here. So the signal sequence and all these proteins which are manufactured in there, they are going to bound to something, or they are going to just fold onto themselves so that the protein buries that portion. And as they are bouncing around in the cytoplasm itself, they will bind to some of the residues of the translocon. So if you look at in your book, it says that there is no involvement of the SRP. There may be some chaperone proteins. We do not know. That could be your graduate work if you go to grad school. So that's serious. I mean, like, if your book says that scientists do not have a clear idea that how that hydrophobic sequence find out the translocon complex without the SRP. It's not known yet, but there could be some involvement of some other structures, but it's not known. So if you read your book, you will see that. Good question. That's a very legit question, but something we cannot answer still. So the next one is the insertion of the membrane proteins into the ER. So far, these were all soluble proteins nicely folded in the lumen. Now we insert them into the membrane. So if you now look at this topology of the membrane, we talked about these multi-pass, single-pass, all of these proteins. What we did not talk about, that how they are in the membrane. So for an example, if we just look at this individually, this is an insulin receptor. And just one thing here, very carefully remember from your previous part. The cytosolic part of the ER membrane, even it is cytosolic, that's perfectly fine, no problem, if it is transferred to another organelle. But the lumen part, if the protein goes out of the cell, the portion that is towards the lumen, if this goes onto the plasma membrane surrounding the cell, this will be the exterior part. Because think about that this is your structure budding out, and then right here, this will come out like this, and it will fuse like this. So the luminal part will be inserted towards the membrane. So that is the most important concept, that the part that is inside the lumen, that's going to be the exterior or the exoplasmic face of the plasma membrane. So that's very important. If it is a vesicle, there's no problem. Lumen side, cytosolic side, cytosolic side. But if it goes on the exterior plasma membrane, then the lumen side is going to stick out of the cell. So here, if we just look at the topology of these, you have a single pass transmembrane domain, although this works as a dimer. You have the N-terminal sequence towards the exterior or the lumen side. Transferrin receptor is on the other way, is opposite. See that here, the C-terminal is the cytosolic side, where the N-terminal is on the luminal side. Here it is the opposite. You have the N-terminal towards the cytosol, C-terminal on the other side. Here you can see that cytochrome, C-terminal on the other side, but most of the protein is towards the cytosolic side. This is the opposite. So they come in all different orientations. And if you see that the multi-pass protein like GLUT1, which is a 12-pass transmembrane protein, the N-terminal and the C-terminal end up in the same direction. And if you just easily can calculate that if they pass through the membrane an even number of times, and we're going to repeat that, you do not have to write. If they pass through the membrane even number of times, like 6, 8, 10, 12, the N and the C-terminal are of course going to be on the same side. But on the other hand, if you look at this one, like a beta-adrenergic receptor, you have 1, 2, 3, 4, 5, 6, 7 pass. Odd number proteins, N-terminal and C-terminal, going to be on the opposite ends. So anything like a 3, 5, 7, like that. So just make sure that you have this basic idea. So once we have that, we have the membrane protein types, and it's like a busy, really super busy slide. This is directly from your textbook. So we have many different types of protein, and depending upon their topology. So let's first take a look at the first, type 1. The type 1 is very similar to the secretory protein. It does have the signal sequence at its N-terminal end. And once that is recognized by the SRP, it will bring that protein to the translocon. It is moving through the translocon. And then what happens, there is a specific sequence which is read right here, which is a hydrophobic sequence. When this hydrophobic sequence is passing through the translocon, it stops the translation for a while. And we call these sequences, we're going to talk about that, we're going to call this as a stop transfer sequences. When this happens, this particular hydrophobic portion is going to diffuse out of the translocon, embed into the membrane. As it moves out of the translocon, then the rest part of the protein is synthesized, and that portion end up in the C-terminal end or in the other place of the cytosol. So we'll again talk about that in detail. Let's have an overview of the idea that what they are. Type 2, it's just the opposite. They don't have a signal sequence. So only the one, which is a type 1, which is closest to the cytosolic, not cytosolic, the secretory proteins which are in the soluble form is similar, but it is membrane inserted. That's the only difference. The whole protein is not going into the lumen. The type 2 is the opposite orientation. N-terminal on cytosolic side, C-terminal on the other way. Then you have a type 3, which is a C-terminal on the other side, and you have just a very small portion of the N-terminal on this side, and this doesn't even have a signal sequence either. Then we are going to see that this one is called a tail-anchored protein. In the tail-anchored protein, the carboxy terminal or the C-terminal is embedded completely into the membrane, whereas the N-terminal is towards the cytosol. And then we have this type 4, which is a multi-pass protein. These are all single-pass, type 1 through 3 and tail-anchored. Type 4 could be two different types. Type 4A, you have even number. Type 4B, you have an odd number. So depending upon the, here you can see here 1, 2, 3, 4, 5, 6, 7. So this is an odd number of pass. That's why the C-terminal and the N-terminal of the protein are two different orientations. If it is an even number, it will go in the same thing. And the last one that we're going to talk about is the GPI-anchored proteins, which also doesn't have a specific signal, but we're going to explain that in a second. So let's start with one at a time. So type 1. this is very, very similar to the one that we talked about previously. So you have a signal sequence, and this signal sequence is cleaved by the signal peptidase, and the protein is translocated as it is being translated. Now what happens that there will be a stretch of hydrophobic residue. This is inside the protein, not at the end, inside the protein, and we call this sequence as a stop transfer sequence. As soon as this hydrophobic residues goes into the translocon, these hydrophobic interactions are going to keep the protein there for a while. As there is no space for the protein to move, ribosome cannot move the mRNA, and it is stuck in that position for a short while. Now what happens from the translocon, that part, hydrophobic part, remember that will interact perfectly with the plasma, with the endoplasmic reticular membrane or the phospholipid bilayer. It will diffuse out of the translocon, and now it is inserted into the membrane, and now it is free of the translocon. And here now the translation will resume, and the part after the transmembrane domain is going to be synthesized. Now you have the N terminal in the lumen and the C terminal in the cytosol. So this is how your type 1 is synthesized. So now we are going to go into the type 2. So here the interesting part is that there is no signal sequence. Instead, what we have, we call this one as a signal anchor sequence. So what is this signal anchor sequence? As the protein is being translated, it will have a hydrophobic residue which will put this particular section into the signal anchor sequence. Now, before that, what happens here? If you can see right here, this is a nascent polypeptide chain, and all of this portion started into the cytosol. It is a little bit difficult, remember that. This protein started into the cytosol. It doesn't have an N-terminal sequence, so SRP will not bind to the signal sequence. So this portion from NH3 plus to this plus end, this portion is synthesized in the cytosol. Then this signal anchor sequence appear during the translation. At this point, the SRP will bind to the sequence. And then this portion is going to get, will be brought by the SRP to the translocon and transferred to the translocon. So remember that the difference in between, that in the previous section where SRP is involved, the SRP binds to the signal sequence, which was at the very end in the signal sequence. But in this case, this is not at the terminus, but instead of that, this is inside the protein, the signal anchor sequence. This is synthesized in the cytosol, the N-terminal. Then once this, again, I'm repeating this just so that you get that clearly. That signal anchor sequence will bind to the SRP. SRP will bring this to the endoplasmic reticulum, to the translocon. It will embed itself to the translocon. And this portion will diffuse out into the membrane, and the rest of the protein will synthesize. That will go through the lumen, and the C-terminal ends up in the lumen of the endoplasmic reticulum. So this is a very clever way to make this protein in the opposite orientation, where this N-terminal cytosolic part is synthesized. So the key thing that to remember, that the N-terminal cytosolic part with these plus charges closer to the membrane, this is synthesized by the ribosome as a free ribosome in the cytosol. Once this red portion or the signal anchor sequence appear, only at that time SRP is involved and brings the ribosome to the translocon, not before that. That then the rest part is synthesized as usual. So now we are looking at the type 3. And the type 3, you have the N-terminal sequence inside, the N-terminal sequence towards the cytoplasmic end, and the other. So last part, you saw that this is the C-terminal end right here. So here is also very similar. You can see that in this position, what happens, you have the N-terminal sequence that is being made. Then you have an internal stop transfer sequence right here. This stop transfer sequence is going to be bound onto the SRP, and that's going to be transferred into the translocon. From here, it's going to be moved into the cytoplasm right here. But the interesting part is that after that portion, after this is moved into the membrane, the C-terminal is going to be synthesized on the other side. So instead of going through the translocon, the C-terminal is sliding from the side of the ribosome, and that's how it ends up on one side. So this is the type 3 synthesis. Now here is a little bit more difficult one, which is a tail-anchored proteins. And this is the type 3 in the reverse, in the sense that here you have a C-terminal end anchored into the membrane. And the problem, what happens and why this is complicated, remember that if you have an N-terminal or in the middle, there is no problem that you are just synthesizing that. As soon as it comes out of the ribosome, the SRP can come and bind to that hydrophobic sequence and bring it to the translocon. Here, the problem is until and unless the protein is completely synthesized and released from the ribosome, that C-terminal hydrophobic sequence is not going to emerge from the ribosome. So that's the problem. So the whole of this protein is synthesized in the cytosol. Ribosome is not attached with the translocon, or the ribosomes are not brought onto the endoplasmic reticular membrane. So complete synthesis in the cytosol, and immediately as soon as this protein hydrophobic sequence arrive, there are these couple of sequences. These are mentioned in your book, but not in the picture. These are called SGT2, GET4, GET5, different proteins. They will immediately come and cover that hydrophobic sequence. That's because it is it's not a favorable interaction. It's in aqueous environment. Immediately, if an hydrophobic residue comes in, you need to cover, or the whole the thermodynamics is going to change in that region. So these proteins now, this SGT2, GET4, GET5, transfer those to this ATP-bound GET3 protein. And the GET3 nicely gets that hydrophobic residue inside that. Now, GET3 is like SRP, but this is not happening while the translation is being made. So GET3 now brings that protein and binds to its receptor, which is GET1 and GET2. And this GET1 and GET2 is not a translocon, because remember that the function of the translocon is to move the protein through the channel into the lumen. Here is no function of this translocon. So the GET1 and 2 is just a receptor to bind the GET3, and this protein will diffuse from GET3 to GET1 and 2 and eventually into the membrane. And here the hydrolysis of the ATP to ADP will release the protein onto this the ADP GET1 and GET2, and then one ADP is released from the GET3. The GET3 is released from the receptors, and the protein diffuse out. So here the complete C-terminal end is buried into the phospholipid bilayer. There is no tail that is sticking out, and that's the most important part. So complete cytosolic synthesis. So here we are looking at the synthesis of a translocation of a type 4, and it gets a little bit more complicated. So here what happens, so in this process, what you have, that there is something which we call as an internal anchor signal sequence. So once this, so N-terminal, there is no problem. The N-terminal is synthesized in the cytosol because there is no N-terminal signal sequence, and that's the most important parts. If you do not have any N-terminal sequence, no SRP involved. But as soon as that part, which is the purple part, internal anchor signal sequence appears, SRP will bind to that and bring the ribosomes to the endoplasmic reticulum. And now you can easily see that this will first internal sequence, which is the anchor signal sequence, this is inserted, and this second transmembrane part is made, but this is also a signal known as a stop transfer signal. So again, repeating that, make sure that you understand that the purple part is the anchor sequence. That means it binds to the SRP and brings it to the endoplasmic reticulum and then inserts into the translocon. Then it starts synthesis back again. synthesize the second transmembrane part, which is a stop transfer sequence. At that time, the ribosome stops. It doesn't transfer the sequence after this sequence. So in this position, these two transmembrane domains diffuse out of the translocon into the membrane. Now, again, the ribosome starts synthesizing the next portion. You have the next second anchor sequence made, and again, the stop transfer sequence is made, but only the first two are made contact with the translocon, none other. That is the only important part, because only the first and the second one needed the translocon to insert them into the membrane. Then second anchor sequence appears, then the third anchor sequence appears directly from the ribosome, and from here these are inserted into the protein, and that's how that stop transfer goes on. So this is how gradually each time the sequence is made, the right here, this will be again associated with the SRP, and this will be associated with the translocon. Once this is associated, two are going to move out. And then once again that is done, again after that stop transfer, again the synthesis of the protein is going to be made, and then it's going to again made into these sequences and move out. Question? Anyone? Yeah. So just make sure that you just gradually just understand that the process, that the first one, the purple sequence, the SRP is involved to bring it to the translocon, whereas the second one is stopping the transfer so that they can diffuse out into the membrane, and then the third one is made, and that will bring it to the translocon, and so on. So that's the process of your type 4. So the last one, which is the synthesis of the GPI anchored proteins, and this is actually not shown, but the GPI anchored proteins also do actually have a sequence which is at the end terminus. Do not need that, but it's a different type of signal sequence. So here everything is the same. You can see that the C terminal is buried, and this is made just like the sequence before that we have seen beforehand. So very close to this. Just only the part is that the C terminal is not on the other side. The C terminal kind of ends at the end. That's the only thing. But what the important part is that this is the precursor protein, and it's going to be cleaved by a protein which is called the GPI transamidase, and after cleaving it transferred to a lipid which is the GPI anchor. So here you have a protein anchor. This is a transmembrane domain, and this will go out. This has no function as such, but this transmembrane domain from the protein, it is transferring to a lipid. And the question is that, this I also discussed in the lecture video that is uploaded, that why there is a need to transfer this protein from a protein anchor to a lipid anchor. And there are certain advantages for that. The lipid anchored proteins are much more mobile on the plasma membrane, so they can move in different directions. That's number one. And the second one is that the GPI anchored proteins, these lipids have high affinity for lipid rafts, so they can be targeted towards the lipid rafts. So without that particular lipid, they cannot be moved into that area. So two important things why the GPI anchor is needed, because it's just like it looks like a wastage of energy. It's already anchored, and why it is going to a lipid leaving the protein. The only reason for that is those two, more mobile GPI anchored proteins and targeting towards the lipid anchor. So that's your last part. Now here, this may look a little bit intimidating, but if you just look at the different small pictures which are in the corners, like if you look at these pictures, which is at the corner everywhere, you can compare those pictures with this one. Oh, I'm sorry. With this one, and this will give you an overview of that which type of protein end up where. So here you can easily see that this gives you an idea that this is your N-terminal signal sequence. This is coming towards the lumen. This is cleaved off. This stays in the lumen, which is the N-terminal. Then right here you have the transmembrane signal, which is the stop transfer. And stop transfer means that this portion is going to get stuck in the translocon. I'm repeating this because it's a little bit complicated and we have the time for that. So a little bit of repetition. So it's going to get stuck into the translocon. From translocon, it's going to get diffused out. Once it diffuses out, the rest of the part is synthesized back again. On the other hand, what happened in the type 2, in this position, there is no N-terminal sequence. So as there is no N-terminal sequence right here, this is going to made right here. This is going to be made into the cytosol right here. Then you have this sequence, which is the internal signal anchor sequence. When this sequence appears after these plus signature sequences, this is going to be involved with the SRP and bringing this sequence to the translocon. Remember that SRP was binding to the signal sequence and bringing this to the translocon. Here the SRP is binding to the signal anchor sequence, bringing it to the translocon. Once it is transferred to the membrane, the rest is synthesized. In the type 3, what's happening here, this is the same way that this is also going to get synthesized into the lumen. Not in the lumen, into the cytosol. There is no N-terminal sequence, so SRP is not involved here either. It's going to get this signal anchor sequence, and here the SRP is involved, bringing this to the translocon. And here, at that time, when this is being inserted into the translocon, this amino terminal is directed towards the lumen. Whereas once it diffuses out into the membrane, this plus sequences after the signal anchor sequence will remain in the cytosolic side. So see that how these two differences are making here. Here the N-terminal, the sequence, signal anchor sequence is towards this plus side. That's why it is remaining in the cytosol. Here you do not have the plus signature sequence before the anchor sequences. That's why this is pushed towards the lumen. But this plus sequences or the plus charges of the amino acids are after the signal anchor sequences. That's why this is ending up into the cytosol. So you can easily see that both of these, the plus signature sequence, cytosol, plus signature sequence towards the cytosol. So this is a way to remember that. And the type 4A, 4B is the same process. There is no difference with that. If you have the plus sequences before the stop anchor, signal anchor sequence, it's ending up with the cytosol, and then it's going with the stop transfer anchor sequence. Again, another signal anchor sequence. Before that, you have a plus charge, and it's ending up with a cytosol. And so on, it will go on. So you can easily see that depending upon that whether the plus amino acids are before the signal anchor sequence or after the signal anchor sequence, that determines whether they are going to be in the lumen or on the cytosolic side. And this is a smart way to do this. It may take a little while, and because you are not reading the book or if you have not seen the video, do not get intimidated by that. If you just read and carefully look at this things, it will make it is very clear. It's not that difficult as such how it may look like now. So here on the other hand, you can see that type 4B, you have the sequence in the lumen, and here both of these end up in the same area, so that you can see that after this signal anchor sequence, if you have plus sequence and you have a plus sequence, and then you have a signal anchor sequence. Here they are all on the same side. So you can easily appreciate that if you have a 1, 2, 3, 4, even number of transmembrane passes, this amino terminal and the C terminal are going to end up in the same direction. And here is the thing that as you have changed this direction, you have 1, 2, 3, 4, 5, 6, 7. If it's an odd number, as both of these are towards the cytosolic end, the orientation of the N and C terminal are going to be on the opposite direction. So this is basically that how the topogenic sequences determine the orientation of the ER membranes. So here we are looking at what is called a hydropathy or the hydrophobicity profile. So you have these thousands and thousands of protein in the database, all these genome sequences, and you pull out a specific protein that you do not know or the scientists have not worked onto that. And if you just plot its amino acid sequence, then you can have something which is known as a hydropathy plot or a hydrophobicity profile, which tells you that some characters of the protein. So you can easily see that each of these blocks that you are seeing are 20 consecutive amino acid. Each of these marks are 20 of them. So a number of hydrophobicity is determined on the 20 residue. So for each one of them, for each amino acid, you have a hydrophobic index for each one of them. And you average the hydrophobic index for the 20, and then you plot those numbers. And you can easily see you have a hydrophobic sequence here, and the top portion is hydrophobic. Below the portion, the negative one, is a hydrophilic. So all the portion that is in the hydrophilic section should be either in the cytosolic or the luminal side, where the hydrophobic portion are the membrane or signal sequences which can be cleaved off. So here you can see that this is a stop transfer sequence. So this is inserted into the translocon and diffuses into the membrane and becomes the membrane spanning part, completely hydrophobic. Classic example, signal anchor sequence, completely hydrophobic. This is a GLUT1, 12 transmembrane domains, and you can easily see from the hydropathic plot that all these are the amino acid sequences that are membrane spanning and hydropathic. So this gives you an overview of the sequence, how you can determine a protein's nature just from the sequence. So let's do the last two of the. So a tail-anchored protein will have a hydrophobic amino acid residue at its N and C-terminal end, N-terminal end, C-terminal end, only internally close to C-terminal, only internally close to N-terminal. This is a very basic protein chemistry question, though. It has nothing to do with what you learned today. Yeah, that is a correct answer. So you can see that the tail of the protein, it starts at the N-terminal. The protein ends at the C-terminal, and that C-terminal is buried in the membrane, so it's a C-terminal end. So it's really a very straightforward question. All right, so here you may take a little time for this one. So which of the following linear sequence correspond to the protein drawn below? You have the amino terminal towards the cytosol, then you have the transmembrane domain, then you have a C-terminal at the extracellular space. Good. All right, so here just one thing, a couple of you who missed that, just remember it's a very small trick. Wherever you see that plus end, that's going to be on the cytosolic phase. That's bottom line. And that's very easy to do that. So, all right.